The brain and eye dissections. The brain, Part 1, lateral view, whole brain. Materials: dissecting tools, preserved sheep brain with dissecting tray, large beaker with water. Note that each lobe is found in each hemisphere. Identify the frontal lobes, parietal lobes, temporal lobes, occipital lobes. Look at the outer surface of the brain. Notice the convolutions and fissures. Also notice that the cerebrum is divided into the left and right sides. Before continuing, sketch and label all 9 bolded structures. Complete all analysis questions. The brain, Part 2, lateral view, cross-section. Make a gentle incision through the corpus callosum to separate the hemispheres. Identify spinal cord, cerebellum, medulla oblongata, pons, midbrain, thalamus, hypothalamus, pituitary gland, corpus callosum, cerebrum, small olfactory bulbs. Complete any remaining steps of your lab and answer the analysis and conclusion questions. The eye. Materials: dissecting tray and tools, newspaper, beaker with water, preserved cow eye. Examine the fat, muscles, and optic nerve. Remove as much of the outer fat and muscle as possible. The eye, cross-section. Apply light slicing cuts as you cut the eyeball in half. The cut should be about halfway between the cornea and the optic nerve. See the diagram in your handout. Identify the vitreous humor. The eye, posterior rear chamber. Sclera. Retina. Choroid. Blind spot. Fovea centralis. The eye, anterior front chamber. Identify the central lens, aqueous humor, pupil, iris, cornea. Ciliary muscle, suspensory ligaments, observe the functions of the lens and the aqueous humor. Complete any remaining steps of your lab and answer the analysis and conclusion questions.